all. Today we shall discuss with some of the important organelles. Among them, mitochondria is one of the organelle which is very important. It is also called as the powerhouse of the cell, as you have already studied in the previous classes, because it is in this organelle that oxidation of food takes place and release of energy in the form of ATP. So let me show you the diagram here. This is the structure of the mitochondria. As you can see, the outer membrane is smooth and continuous, while the inner membrane is thrown into finger-like projections called cristae. This cristae that increases the surface area of cellular metabolism, that is oxidation. Now, as you can see in the diagram, the inner membrane consists of stalk structures which are called as oxysomes or elementary particles. It has a head, this middle part, and the base. Here and the lower part contains the components of electron transport chain. Clear? Now the matrix, which is also called as mitochondrial matrix, consists of ribosomes as well as organic and inorganic compounds, and most important, a circular DNA which is called as mitochondrial DNA. So just like chloroplast, mitochondria is also referred as semi-autonomous organelle since it can synthesize its own mitochondria. Next important organelle is the endoplasmic reticulum. Clear? There are two types, rough endoplasmic reticulum and smooth endoplasmic reticulum, shortly called as SER and RER. What is the difference? Endoplasmic reticulum, it is called as rough endoplasmic reticulum because it has ribosomes on its surface. Where it is located? It is located near the nucleus, on the nuclear membrane, and it is involved in protein synthesis. While the smooth endoplasmic reticulum, you can see the tubular structures, they are involved in the synthesis of steroids and lipids as well as in their transportation. Clear? Now when we talk about one more next organelle which is very important are the lysosomes. Clear? So in the lysosomes, what you can find? These are rounded structures, spherical structures which are found only in animal cell. First discovered by the Christian D. Wave. Clear? They originate from Golgi bodies. We had discussed in the previous class that the Golgi bodies are made up of cisternae, that is, and vesicles and vacuoles. So these vacuoles or the vesicles, they get pinched off and form the lysosomes. Lysosomes are bounded by a single membrane and they contain about 40 hydrolytic enzymes such as proteases, carboxylases, lipases, and so on. So lysosomes, there are four types: primary lysosome, secondary lysosome, residual body, and autophagic bodies. Clear? Primary lysosome give rise to secondary lysosomes by the addition of extra materials, and they are involved in digestion. Now, when we talk about residual lysosomes or residual body, after the digestion, the leftover part is left in these residual bodies and they are thrown out by the process of exocytosis. Now, the fourth type of lysosome is autophagic type, in which what happens, where sometimes the cells get old, worn out, or injured or damaged. Then in that case, what happens? The lysosomes secrete autolytic enzymes that can digest or phagocyte its own cell. Therefore, lysosomes are also considered as suicidal bags. Now, when I talk about mitochondria, it also performs some other functions besides cellular oxidation. It helps in yolk formation, and in the human sperm, the middle piece is contains mitochondria that helps in the movement of the sperm, and it also is involved in apoptosis or programmed cell death. Clear? Beside this, you also have the term called as endomembrane system. What is that? Organelles such as endoplasmic reticulum, Golgi bodies, vacuoles and lysosomes are called as endomembrane system because though they are unique in their structure and function, yet they coordinate with each other or interconnected with each other. Therefore, they are referred as endomembrane system. Next type is the cytoskeleton. Clear? Cytoskeleton that involves the microfilaments and microtubules. So microtubules, these are tubular structures usually around 13 in number, and they are made up of the protein tubulin. They are arranged in concentric pattern 9+2, that is doublets of tubules along the periphery, and the central part consists of a pair of tubules. They are you can say they are hollow and unbranched, whereas when we and are usually involved in the formation of spindle fibers during cell division, formation of cilia and flagella. Clear? Whereas when we talk about microfilaments, these are solid, unbranched, shorter than microtubules, made up of the protein, actin and myosin. Clear? They are usually involved in providing support. That's why they are referred as cytoskeleton. Beside this, you have microbodies which are called as peroxisomes, sperisomes and glycosomes. Perioxisomes, they are small spherical vesicles. Clear? And they are called as microbodies because they are very small. And when we talk about peroxisomes, they are involved in hydrogen peroxide, that is peroxidase synthesis. Got it? In the presence of enzymes called as catalases, they break down harmful hydrogen peroxide into harmless water and oxygen. Similarly, we have got glycooxysomes which are found in fat-rich plant cells like in the endosperm of the castor seeds. So these peroxisomes or glycooxysomes, you will be asked in 1 marks or 2 marks questions. Where are they found and what is their function? Clear? Lastly, we come to the organelles such which are called as cilia and flagella. Clear? We have discussed in the prokaryotic cell also. Here also, cilia and flagella, they usually perform the function of that is movement. Clear? Either in undulating movement or in wave-like motion. Clear? Let me show you the diagram given here of the you can see the central core which is called as axonome. Got it? And these are the doublets of tubules in pairs which are 9 in number. Clear? And a pair. So this arrangement is referred as 9+2 array. Clear? Now let us come down to the notes. Mitochondria. Was discovered by Colliker, but the term mitochondria was given by Benda. It consists of outer membrane, inner membrane and mitochondrial space also. Then we have, after mitochondria, mitochondrial matrix, the ground substance, as you can say, consists of enzymes of Krebs cycle, which is also known as TCA cycle. We will be doing in detail in the chapter respiration, cellular respiration, as well as they contain ribosomes, own circular DNA. Then we have the endoplasmic reticulum. So you apart from the contribution which I have given, you may be also asked who invented or who first discovered the different organelles. Like lysosomes was discovered by Christian Dave. Why it is called as suicidal bags? What do you mean by residual bodies? Endoplasmic reticulum, questions can come as difference between smooth endoplasmic reticulum and rough endoplasmic reticulum. They are tubulars form. And then you have lysosomes. They originate from the Golgi bodies, absent in plant cell. That is also one of the features of the difference between plant cell and animal cell. Presence of lysosomes, absence of cell wall, and presence of centrosome with centrioles. In the centrioles, though we have discussed earlier, the arrangement is 9+0 array, and they are made up of the protein radial, after the lysosomes we have the last part, cytoskeleton. This part you have done, second part. And the diagram, I think, diagram. So best of luck for your upcoming board exams, since this was the last session before your exams. After the exam we'll again resume with the tutorial classes or video classes. Stay home, be safe.